Welcome, everybody. Today, we will be having a discussion around vaccines and the development of vaccines specifically for the treatment of COVID-19. As part of the IPAC conference, this will be a discussion panel format. My name is John Pisatsky, and I will be the moderator for this discussion. Before we get into the important questions around vaccinating Canadians for COVID-19, I would like to first start out with a primer to initiate our discussions around the important questions. I've prepared a short presentation here to get everyone up to speed on some key concepts and to remind people about the greater context of COVID-19 as a pandemic of modern times. So first, I would like to introduce to you this slide, which comes from the website listed at the bottom of this slide. And I'm presenting this to you to show the impacts of pandemics through the course of human history. And important to note that there is a constant barrage of pandemics that humans have had to face over a 2,000-year history that remains constant as a challenge for humanity. So we've had huge numbers of deaths associated with pandemics, starting from basically the beginning of recorded history. And these pandemics have had untold human costs. As we develop medical and biotechnology solutions to these infectious diseases, the burden of these pandemics is less, but they are a constant concern for us as researchers and you as policymakers. So as you can see, we've had a number of pandemics even in the last 50 years, including SARS, swine flu, Ebola in 2014 to 2016, MERS, and the topic for today's discussion, the COVID-19 pandemic, which is still ongoing. These pandemics that originate through either bacteria or viruses cause an untold human death toll. And it's the job of vaccinologists and members of our expert panel to find solutions to lessen that burden and to find ways to protect our population through vaccines and therapeutics. This is not a new problem, and Canada has been considering infectious diseases since before the founding of Canada as a country. And we've had a number of important positive steps that have improved Canadians' health. Too many to discuss here, but if you're interested, the Canadian Public Health Agency has a very good summary of this. It's important to think about and remember that we were considering vaccine strategies as a way to protect the population as early as the 1800s in dealing with smallpox epidemics. We've had a number of successes in terms of developing and deploying vaccines in Canada, which ultimately has improved the quality of life and the increased mortality of Canadians. So before we get into a detailed discussion, I want to show you the basics of vaccines. When we think about a vaccine, we think about the virus, in this case, coronavirus. SARS-CoV-2 is the virus that causes the symptoms of COVID-19. And we think about it as this sphere here with these spike proteins on the surface of it. The architecture of a virus is in this case, is an envelope virus with spike proteins and an RNA molecule, which is the genome of the virus in the center. This architecture is important when you think about and talk about vaccines, because the spike protein, this outer protein that forms the casing for the virus, is one of the key targets for making vaccines. If our body can recognize this protein, then and defend against it, then we can defend against the virus in total. When we think about how the coronavirus works in very simple terms, it enters the body, it enters lung epithelial cells through interactions with a receptor, and then the virus genome is released and translated into new proteins, which catalyze the virus life cycle and produce new virus. When we think about vaccines, we're trying to stimulate the immune system and to develop an immunity. We say immunity, but what we mean is that the body will recognize an invading pathogen and deal with it quickly and efficiently before it becomes a health problem to the individual who's exposed to the virus. So how this part works is that the virus, as it's replicating, stimulates an immune response. In a simplest terms, it gets recognized by a cell that produces antigen. The antigen is recognized by various cells of the immune system, some of which generate antibodies. And those antibodies then are part of the battle against the virus, and they recognize, as again here, the spike proteins that are surrounding the virus. Various cells of the immune system then, in conjunction with the antibody response, can destroy the virus by various mechanisms. And the importance is that, an important point is that these immune cells, these blood cells that are circulating in your body, can retain memory of this pathogen. And it's through this memory that you have protection. So what kind of vaccines are being developed currently to treat SARS-CoV-2? Well, there's a number of different categories of virus vaccine strategies, some of which involve weakened or inactivated SARS-CoV-2 viruses. And in this case, we're teaching the immune system by giving either an attenuated virus or an heat-inactivated virus, which is non-infectious. We're training the cells of the immune system to recognize, for example, the spike protein, and have this wall of defense created. In other cases, there are technologies that are what we call nucleic acids, like DNA or RNA. And in the recent press release from Pfizer, their successful vaccine or supposedly successful vaccine, which provides 90% protection, is actually an RNA vaccine. And this RNA is injected into the human body. It enters into cells, and the RNA contains the genetic information to produce the viral protein spike. And that spike protein then generates an immune response. So the nucleic acids carry the genetic material and then cause the cells of our bodies to produce the proteins of the virus, which generate an immune response. We can go further and just give patients the protein itself, and that is a class of vaccine we call protein-based. The protein-based vaccines can be through viral vectors, or it can be purified proteins or protein subunits, or they can be virus-like particles that are injected into a patient, virus-like particles being a type of vaccine that's being developed in Canada by Medicago. 4 or 5 months ago, as of 4 or 5 months ago, there's hundreds of research efforts being conducted to develop vaccines in each of these types of categories, virus, viral vector, nucleic acid, or protein, or other new technologies for vaccine strategies that are in the research to clinical trial phase. It's these vaccines that potentially will give us resolution or at least some relief to the current COVID-19 pandemic. And it's this research and development that is critical to our rapid responses to future pandemics. And this is part of our discussion today with our expert panel members. So it's my pleasure to introduce to you all our expert panel, which is comprised of several of leading experts in virology and vaccine development within Canada. And first, it's my pleasure to introduce to you Michael Houghton, Professor Michael Houghton, who's the director of the Li Ka-shing Institute for Applied Virology at the University of Alberta, and our most recently minted Nobel Prize winner in Canada for the discovery of the hepatitis C virus. Michael Houghton is an expert on developing both vaccines and therapeutics against viruses, and we're honored to have his presence today for our panel. Second, we have Alison Kelvin from Dalhousie University, who's actually currently seconded at the VIDO Vaccines and Infectious Disease Institute in Saskatoon, and she is an expert in respiratory viruses and the development of vaccines and therapeutics against respiratory viruses, viruses that target our lungs and our respiratory system. Third is Professor Lauren Tyrrell, who's a founding member director of the Li Ka-shing Institute for Virology and distinguished professor at the University of Alberta. Professor Lauren Tyrrell is an expert on viruses that cause hepatitis B virus, hepatitis C virus, and is both an entrepreneur and a clinician, as well as a researcher. He has made distinguished contributions to the discovery of therapeutics against hepatitis B virus, as well as making important contributions to hepatitis C virus as well, and is currently also a member of the COVID-19 vaccines task force in Canada. Lastly, but not least, is Professor John Bell, who's a senior scientist and professor at the University of Ottawa and leader of the Cancer Therapeutics Program at the Ottawa Hospital Research Institute. Professor John Bell is a virologist and an immunologist and also an entrepreneur. He's made his name in developing viruses for treating cancer, what are called oncolytic viruses, but is also currently involved in developing vaccine strategies against the SARS-CoV-2 virus that causes COVID-19. So with that, I would like to get going with a series of questions for our panel to discuss. We have a lot to unpack, so let's get to it. Our first question for our panelists to discuss is a very basic question. Why are vaccines successful? What makes a vaccine succeed or fail? And what does a percentage of protection mean in real terms when we talk about vaccines for treating various infectious diseases? And I think we'll start off our discussion with Professor Michael Houghton.
Thank you, John. I think usually vaccines fail because of lack of efficacy, that despite animal studies showing that they work and other preclinical data supporting efficacy, in the real situation of human vaccination and follow-up, usually the reason for failure is insufficient efficacy. You may only see 10, 20% efficacy or zero. And you never really know until you go into humans with the vaccine to determine efficacy. This is why the regulators require you to prove efficacy in the human population rather than approve vaccines based on animal studies. Now, the issue of threshold is somewhat complex. In terms of COVID, for example, obviously, ideally, you would like a vaccine that prevents infection. Nobody will get infected at all following the vaccine for the rest of their lives. Unfortunately, that ideal is rarely met with human vaccines. And so usually what is most important is that a vaccinated person will not get disease when they encounter the virus in the future. They may have transient infection that you can detect, but you don't want them to get disease and you certainly don't want them to die. So I think with COVID, regulators are using 50% as the minimum threshold for efficacy. And I think that mainly will mean that 50% of disease is prevented, 50% of deaths, and hopefully 50% of infections. But I suspect even if a vaccine was shown to be less efficacious than 50, for example, 30 or 40, provided it's decreasing disease and decreasing fatality rates, I suspect it would be approved if there was nothing better around in terms of another vaccine. So in general, up to now, vaccines have, the baseline has been considered 60 to 70% efficacy, that you actually prevent disease in around 2/3 of the people you give the vaccine to. COVID, because of its severity, already the regulators are saying, well, we'll accept even a 50% efficacious vaccine. And like I said, they may even accept a lower level of that. Now, what we heard a few days ago from Pfizer and BioNTech using an RNA vaccine to COVID is that within their follow-up period, which is quite small, we have to realize is only 1 to 2 months, they're estimating an efficacy of 90%. So that is wonderful news. It's really encouraging. And we just hope that over time, that vaccine is shown to be very efficacious in the long run and not just in the short term. But it's a very great start.
I think it's great points, Mike. I think another concern in the vaccine world is that with some viruses, and it's not been wholly proven with COVID yet, but some viruses, if you have too weak an immune response elicited by a vaccine, it can actually be counterproductive. In fact, you can make the disease worse. So it's you're absolutely right. You want to get a certain level of protection, but you also have to be careful that you don't have a vaccine which is too anemic, as it were, and actually can make it a whole lot worse than it is currently.
It really varies with different viruses we're looking at. I mean, some vaccines are highly effective. For example, the polio vaccines, or a number of them are near 100%. But it really is some of the respiratory viruses where we're looking at rates of protection that can vary from, as you said, 20% to 70%. And to see this new vaccine result from Pfizer in the 90% is quite unprecedented for respiratory viruses at this stage. And it bodes well if that can be held up over time.
I agree. It probably depends on the virus. If we look at influenza or HIV and their high mutation rates, that's really become a problem to developing effective vaccines, whereas other viruses such as polio are more easily targeted.
Going back to the COVID, it's worth mentioning that this is a very large RNA virus, and large RNA viruses, usually RNA viruses don't get much bigger than about 10,000 bases. This one is 30,000 bases, and it replicates okay, but it's unique in that it has a protein within its virus that does proofreading. So if you're making mistakes in the nucleic acid, there is proofreading, and that may give more stability to the virus, that our vaccines may be more effective because of that proofreading activity and less mutation.
Wow, that's great. Great discussion around that point. Let's move to the second general question. What have been some challenges in designing a COVID-19 vaccine? And is there anything about the designs that make deploying the vaccine particularly challenging compared to other viruses like what we've been discussing so far?
Yeah, so I think that's a great question. And if we think about SARS-CoV-2, really, it's an emerging virus. We had no idea what this virus was, in December last year. So we're starting from the ground up. We were lucky that we had two things on our side. One is the quick sequence which was delivered for the virus. So we were able to identify that. And then the second is that we were able to see similarities with this newly identified virus and the original SARS from 2002, 2003. So that enabled us to perhaps have some idea of how the properties of this virus would behave by comparing it to the original SARS virus. But really, because it was an unknown virus, we had to start again from the ground up. We had to identify how the virus would infect cells. So what was its targeted receptor? We had to identify an animal model that was susceptible to the virus so that we could start evaluating therapeutics and vaccines. We have to identify, and we still haven't done this, a correlate of immunity for what is protection after you're infected with this virus, and which is important for determining how effective a vaccine is going to be. So I think considering this, our major hurdle was that this was a brand new virus. But then when we think about SARS-CoV-2 as a respiratory virus and developing a vaccine for this, it's been well documented that vaccinations for respiratory viruses through an intramuscular injection, so an injection into the arm, aren't always as effective at protecting infection within the upper respiratory tract. So this is where the SARS-CoV-2 virus is replicating in many people and how it's transmitting. Whereas we find more effectiveness in a vaccine platform specifically for SARS-CoV or for respiratory viruses when the immunization is given within the respiratory tract. So taking this together and thinking about how to distribute this vaccine when we haven't even determined what is effective, we have a lot of hurdles, I think, in the future to overcome.
Mm-hmm.
Yeah, I think another thing that we're still, and I think Allison's spot on, we don't know enough yet because this is too new a virus. But, with the SARS-CoV-1, which is the first SARS virus that we had a big issue with, it looks like the long-term immunity might be related to your immune cells, T cell responses, as opposed to the antibody response for long-term protection. And so that may be true in this virus too. We don't know yet. And I think we're going to have to see, when you design a vaccine, you want to make sure maybe that it gets both arms of the immune system activated. And I think that's a question we still are up in the air about.
And I think that relates a lot to the adjuvants that are being used with these vaccines. And we are seeing different ratios of antibody to T cells with different adjuvants. And I think some of our long-term immunity and long-lasting immunity may vary with the adjuvant that is being used.
Well, I certainly would agree with Allison that for respiratory infection like COVID, you want to really design a vaccine to have strong mucosal immunity, that you design it to give you very strong immune responses in the nose, in the throat, upper respiratory tract, and lower respiratory tract. It's interesting that most of the approaches so far are not specifically designed to do that. The RNA vaccines, the one reported by Pfizer, It's given intramuscularly as an RNA, obviously, and the RNA is translated into the protein, so the body sees it as a foreign object and makes an immune response. And thankfully, it seems to be working after 1 or 2 months, which is great. But there are other ways, as Alison indicated, that you can activate mucosal responses in the upper and lower respiratory tract. So we have a lot of methods to try against COVID. We're just hearing about the first results with one RNA vaccine, which is very encouraging, but we have a lot of others that we can try to even increase protective immunity.
Wow, this is really interesting and some great discussions. I think this is a good segue into our next question, which simply asks, is there a leading technology that we think it might be the preferred method for immunizing for COVID-19 in Canada? And we've heard about the first successful clinical trials, but is that really ultimately the strategy that we want to follow for worldwide immunizations?
Sure, you're going to get a biased position from me because I'm a virologist, and so I have a certain bias to how you should go about doing this. As you've mentioned, the RNA-based vaccine, which is basically you take genetic information and synthetically make that and inject it into a person. As Mike said, it then makes the viral protein there that your body reacts to. And what it's trying to do is it's trying to mimic a virus infection to some extent, so that your body will react to that and start to respond and make an immune response. And it's also true of other kinds of vaccines, something we call a subunit vaccine, like the Medicago one, where you take a piece of the virus and you use that as a way to immunize, maybe with an adjuvant, as Lauren suggested. And again, you do what's called viral mimicry by making it look like a virus infection. My position would be, why not just use a virus? Because it looks more like a virus right there. You don't have to mimic anything. And there's multiple ways to do it, as you mentioned, John, earlier. One is to take pure SARS-CoV-2 virus and use that, but kill it first so that it's actually a dead virus, can't really spread, and inject that. And that's been used in other vaccines like the polio vaccine as an example. But another way to do it is to use virus-based vectors, which go in and are benign on their own, but they can replicate a bit and have them express SARS-CoV-2 proteins. So they express it in the context of a virus infection. And so therefore, you're going to have a really good, robust response to it. And so there are a number of, and some of the most successful vaccines have been of that sort. For instance, we've all been vaccinated with mumps, measles, rubella. And that's a way to, and those are all live viruses which replicate a little bit and you make an immune response to, and it's a very successful vaccine. So, I hope that the Pfizer one continues to work great, and I hope we have long-term immunity from that would be ideal. But if it doesn't, I think we need to think about other kinds of vectors like virus-based vaccines or subunit vaccines to come along. And maybe what we really need to do is have the Pfizer vaccine followed by a boost with another kind of vaccine. So maybe that's the way we're going to ultimately get the most robust immune response to ensure that we can defeat this virus. So it's interesting that we have lots of choices to test. And I think in the field, everybody agrees, we don't know the answer yet. We got to try a number of things, get a number of things to the clinic to find out which one or which combinations are going to actually be the most potent in fighting this pandemic.
Really does show that we need to examine different forms of immunization when we have a major disease like this and pandemic, because we're not sure which one will give us the best results. But careful monitoring and using, we've proved in Canada have looked at international vaccine candidates that are messenger RNA, there's adenovirus delivered, and protein adjuvanted vaccines, as well as the Medicago virus-like particle. And these, it'll be very interesting to compare the results from these and carefully analyze them so that we know which one is going to give us the best results and how will we modify vaccines going forward so we can come up with even better protection.
I think we cannot emphasize enough what good news we received from Pfizer and BioNTech a few days ago. It's hard to imagine how we would be feeling now if that had failed. So I think we really are incredibly encouraged by that work. But I agree with John that, what we want now, we're greedy. We want long-term immunity. We want people to get a vaccine and they be protected for several years at least. And I agree with John that probably that's going to involve mixing a combination of different vaccines, a combination of the adeno-vectored vaccine, as he mentioned, along with an RNA vaccine, or along with the subunit vaccine that Lorne mentioned. So these are all things we've got to figure out in the future. But it's just so wonderful that the Pfizer data looks so good. And we have so many technologies that are being applied to COVID. We mustn't forget that it's not just a question of which is an efficacious vaccine. We've also got to consider the logistics, and perhaps Lorne can deal with this better than me. They're not all easily available to the population. Lorne, you probably want to say something about that.
No, coming back to the Pfizer vaccine, it is it's wonderful news that this has shown such good protection, but everybody's cautious because it's very early on. I mean, this study of 45,000 patients in the phase 3 study is really looking at an interim analysis, and that was released, and it's releasing good news. But we need to see the full study completed, and we need to see longer term protection. I think that's very important. We also need to see if there is any evidence of breakthrough or resistance viruses that will escape this immune system. But the key for particularly the Pfizer vaccine is that there's two things to vaccine. One is the vaccine substance, which you can produce in a lab. And then there's a vaccine product, which goes out to the public health people and is able to be injected into patients. And the Pfizer vaccine has the issue because it must be maintained at minus 75 degrees. And that cold chain, even in wealthy countries, is going to present some challenges. It's not easy in public health offices or doctors' offices or pharmacies where you might be thinking you're going to get your vaccine easily. There is an issue of going from the substance to the product, and that product has to be delivered in minus 75 degrees out to the sites. So I think we have other vaccines that are coming that should be much easier to deliver, and we'll see what their protection rates are and how that will be. But it is a key issue with the Pfizer vaccine.
I really agree with the point that we need to be backing several of the technologies, especially considering the number of people that we need to vaccinate globally, and just even in our country, that we might have supply issues if we put, say, all of our eggs in one basket. So I think the more vaccine that we can and safe and effective vaccines that we can have, the more people will be able to vaccinate. Aside from that, I'm really excited about the mRNA vaccines, especially considering emerging viruses. It's estimated that for a vaccine to be effective at preventing a pandemic, we'll need to get vaccines deployed within 4 months of identifying an emerging pathogen. So I think the nucleic acid vaccines really may offer a solution to this because of the speed. Of course, I understand the importance of keeping viral antigenicity, keeping the virus looking the way a virus should look to have an appropriate immune response. But then there's been some issues around that, especially looking at the original SARS vaccines, the formalin inactivated vaccines possibly triggered an inappropriate response. So something that we're keeping in mind here right now as we're evaluating SARS-CoV-2 vaccines.
So maybe I'll let you, Lauren, speak to this. What should Canada be doing? What is Canada's plans currently? What should Canada be doing in terms of developing a policy for the rollout of vaccines or a vaccine or vaccines to treat the Canadian population, and how should we be monitoring them?
Well, these are very good questions, and Canada is unique because Canada has health deliveries under the provinces, and the whole thing we've done on the vaccine procurement and which ones we should get has been done primarily by the federal government. Then they moved this into the public health services, and the public health services moves it out to the provinces and the territories. So the provinces and the territories will have to be heavily involved in how we roll out this vaccine and how we monitor and who will get it. It is, I just want to say that I think on the procurement, Canada got some criticism in the beginning because we might have been behind, but I think that using scientific advice was one of the keys that the Canadian government did. The two ministers from Innovation Science and from Health asked for the committee to be set up in the vaccine task force. There are a number of advisory committees to them, but what I'm stressing is they have used science to give advice, and it is not politics that's interfering with the advice of scientists. They've used that advice, and I think that's really very important. We have, Canadian, the National Advisory Committee on Immunization that will help decide who will get the vaccines initially. That's been their role, working with public health, giving advice. There is also the Therapeutics Task Force that looks at therapeutics. There's the COVID Vaccine Task Force, and there's an Immunity Task Force. And all of these will be involved in how some of the monitoring will be done as these vaccines roll out. And I think there'll be pretty close monitoring of how effective the vaccines are, how long the immunity will last are key questions, and will we need boosters or not? This, I think, is being set up very well through these various committees. And there has been good funding for those committees so that administered through those committees, so that we should have the ability to do this in Canada quite effectively. The big issue will be the rollouts. Some of them will be much easier to roll out, and if they have high protection rates, that'll be wonderful. But for what we're looking at, the current one, when people say, when would people get access to this new vaccine? It'll probably be in Q2 of 2021 before there's good access to the Pfizer vaccine. It may be available sooner, but when you say when will the public get access to it, probably in Q2.
I'll stick my mouth out there again because I do have another bias. That is that, there are some groups that are at higher risk, and one that I'm very concerned about are cancer patients. We know from China that cancer patients got really battered by this virus. And so, I'd like to see cancer patients be high up on the list of some of the first people to get vaccinated if possible.
But you're going to see people over 65, healthcare workers in the front lines, and then people with comorbidities, which I guess would include the cancers, given the results that are worse with COVID in cancer patients. Yeah.
I don't, Lauren, what do you think about some of this stuff where not even a specific vaccine, but, for instance, vaccination with BCG seems to train the innate immune system to respond better. Is that something that's being considered at all as a strategy?
I don't think we have considered it at the vaccine task force, but I'm not sure that I feel well enough versed in that to give good advice on whether BCG should be used in these.
Yeah, I think that those are good points. There's a lot to consider. The WHO, as well as ACIP, which is basically the American version of NACI, have come up with a good prioritization plan that is based on a couple of phases or pillars. One, comorbidities and people with at high risk for developing severe disease. The second would be people at high risk because of their workplace or their living arrangements. So I think we have to start thinking about underserved communities here, people in remote areas where if there was an infection to happen, it would easily spread throughout that community. And then also communities maybe in inner cities that might be overlooked, like homeless shelters. So I think that there's a lot to think about, and then the logistics of actually getting it to these people, not just who should be prioritized. So making sure that they're equitable across our country, and then also how can we participate in a global equitable delivery of vaccines. So I know that the WHO has been working on this with several countries. I'm not sure where Canada stands on this.
Well, Canada is a member of COVAX. It means that we are purchasing vaccines that will also be distributed to countries that are in the COVAX organization. You know, we believe very much that vaccination is a worldwide issue, and if we're going to control this disease, all countries need to get access to the vaccine. So Canada is in that. We're doing our part in that regard.
Right. And I think that would be a great piece for a rollout plan. You know, if there's how many doses will stay in the country and then how many will be, I guess, shared with other countries.
Now, some of that has already been worked out by the Canadian government in their participation in COVAX and in their procurement. I would point out that, you know, not very far behind Pfizer. First, I would say that Pfizer is putting a lot of effort in how to roll out the vaccine to different sites. They're looking at, they are working to see how it can be distributed to different sites, and including, you know, transfer on dry ice and containers, etc. So it could be done. But Moderna is coming along with another RNA vaccine, and that vaccine requires -20 degrees. And that'll be easier to distribute because really it is freezers, regular freezers. And then behind that, the AstraZeneca and all the adenoviruses of Janssen and AstraZeneca require -2 to 8 degrees, regular fridges. And the adjuvanted proteins all require 2 to 8 degrees. So there will be less problems in the distribution of the other vaccines because the cold chain should be much more favorable.
Yes, I totally agree with Lorne. In a few weeks' time, we're hearing that the Oxford AstraZeneca adeno-based vaccine will have efficacy data. And as Lorne said, if that is positive, if we see very good efficacy, that's going to be a much easier vaccine to distribute around the population. And then potentially the mRNA vaccines, which require a cold chain, which is difficult to distribute, not impossible, but difficult. Maybe they could be used to boost recipients of the adeno-based vaccine. So we're keeping our fingers crossed that we get more good news in a few weeks from the adeno-based COVID vaccines.
So I would like to ask another question of the panel, which is more practical. So now we see the possibility and likelihood, in fact, that we'll see multiple vaccines being distributed and purchased, procured, and distributed. How should Canada monitor efficacy of vaccines and track this moving forward?
Perhaps Lauren would have better insight than me, but I would see that considering adverse events and as well as just vaccine oversight is done through Health Canada as well as PHAC, that PHAC and Health Canada will be monitoring both if vaccines are safe and effective before they go in, and that they can be produced at highest quality before they go into people. And then they continue this responsibility after these vaccines are being administered into people. there is a committee to oversee adverse events. So I would figure that committee would take on this job of making sure that any adverse events after a COVID-19 vaccine administration was recorded and properly analyzed. And then I think looking at what occurs for nationally for us for the influenza vaccine, we have the Canadian Sentinel Practitioners Surveillance Network, which basically associates any influenza medically assisted or medically advised cases and determines the percentage of those in people who had been vaccinated. So I think that would be a nice network to piggyback onto with the COVID-19 vaccine, but perhaps something else has been devised, I don't know.
I think there will be continuing very close monitoring of the results with these vaccines, and that will be part of the expertise of the vaccine task force also, to look at some of this with the immunity task force, as well as the public health agency. So let me just say that it's often worth stressing there's a big difference between a therapeutic and a vaccine. A vaccine is going out to large populations of healthy people, and we monitor very closely for any side effects. If you have one serious side effect in 10,000 people, this usually kills a vaccine. So I think there's a lot of monitoring going on to make sure these vaccines are safe going into healthy people. As a therapeutic, you've got a serious disease, you can put up with a little more side effects and than you would with the vaccine. So I think that's very important to stress. I want to also stress that I think the technologies that we're using, many people say they're new and are they going to have more side effects? In fact, I think the technologies in many ways are cleaner than vaccines of the past, where you might have it contaminated with another virus or a virus-like particle. The technologies today are very clean, and the messenger RNA or the DNA, these are all, I think, much better technologies than before. And I think that gives us more confidence of the safety of these vaccines. The pandemic has really brought along some wonderful technology and wonderful cooperation between industry, academia, and governments. And this has been forced on us by a threat of the pandemic, as we've seen its social impact, its health impact, its economic impact. Everybody feels urgency about working together. And although it's quick, I think the new technologies we're applying in many ways are safer than the older technologies. And one of the things we're going to be facing is vaccine hesitancy, because they are newer technologies. But I think the vaccine hesitancy, we'll all have to address that. It is an important question. You take a vaccine for two reasons: one, to protect yourself, and one, to get community immunity up, or herd immunity. So you're doing it for the community and others, but you're also doing it for yourself. So vaccine hesitancy will be a major problem, and we have to work hard to make sure people feel confident in these vaccines.
I think COVID is such a threat to all of us. And even if you're a young person and a healthy person and you get infected, but you don't get serious disease, you have a real chance of transmitting it to a cancer patient or to an elderly person who will suffer tremendously from getting COVID infection. So I think there's a huge risk of getting COVID infection. And I think because of that, people need to get vaccinated for themselves and for the community. There are several phase 3 trials ongoing around the world with different vaccines. Each phase 3 trial is involving at least 36,000 people. So there will be strong safety indicated by these vaccines, hopefully. And if there isn't, then as Dawn said, they won't be used. But I think we'll be in a situation where the vaccines are well tolerated. There's minimal risk to the person from getting the vaccine, but there's huge risk to the person and to the community if you don't take the COVID vaccine. So I think COVID is an example where vaccines can really, really help yourself and the community.
Well, just a comment on what Lauren said. I think he again is spot on with a lot of this. One that he mentioned is that the severity of this disease has, galvanized us all to work together and to get money out to do this. But it would be nice if that was maintained after this, so we don't have to go through it again. So I think an important message is let's keep working together, but let's keep the funding up for vaccine and all aspects of research, because this has happened, but it's going to happen again. We had already SARS-CoV-1. We all know that was in Toronto. It was a big deal, and now we've got much worse. So I think that can we build on this? Can we build on this and have people say, yeah, we've got to change our funding system and make sure that we have immunologists and virologists who are employed here in Canada, who are going to maintain this momentum that we've picked up on this because of this pandemic.
Could not agree more, John. I think that Canada is part of our work on the vaccine task force is to try to say that how do we build more capacity and capability in Canada and ensure that we are in a better position when the next pandemic hits. But, I think a year ago now, if we'd have said that a small piece of RNA could bring the world to its knees, you would never have many people listen. Mm-hmm. After this, you will understand that it can happen. And, we all expected influenza to do something like this. And, we're looking at the virus, fortunately, that has it still is serious with the mortality rates. And I would say it's also very serious when we see some of the long-term effects of this virus. But we know of examples of viruses that cause a much higher mortality rate. MERS and SARS-1 did, yet these high mortality rates, fortunately, did not transmit like COVID-2 or COVID-19. And if we get a virus that has got a high mortality rate and a high transmission rate, which is possible with an influenza, then we have a huge problem, and we've got to be more prepared. As Alison said, we got to be able to do something within 4 months. I got to say that public health has done a pretty good job of trying to control these, but I think public health in many countries has had a lot of political interference because of the economic impact.
Is there something that Canada should be doing that hasn't been doing previously around funding and continuous funding for research into infectious diseases and in vaccinology that we need to pay more attention to?
Yeah, it's a great point, John, about clinical trials, which are really not well supported in our country, unfortunately. And it's considered that clinical trials are considered more like research than healthcare. But certainly in my field, in the cancer area, that's really not the case. Many patients go on to clinical studies for the care. And so I think we need to change how we fund clinical studies, and actually come up with a plan to fund them, I should say, so that we can actually do clinical studies that are not only just asking a question, but also potentially helping people who, in the case of a vaccine, become vaccinated earlier on, for instance. In the case of cancer, get onto a new experimental therapeutic, which may help them. So I think there's a number of things that the pandemic has shown us where we have weaknesses in our healthcare and research system, and we need to bring things together and have a more coordinated plan so that we do support research and healthcare together, because I think both of them go together very nicely and support each other. But I think right now it's very siloed funding. We don't have a great plan, and there's been a lot of weaknesses exposed, and in the hospitals, for instance, which are suffering terribly in Montreal, for instance, because they don't have the right support and healthcare workers there. So I just think we need to have, as a country, a better leadership in thinking about this problem together so that we don't come to this again. And again, fundamental research and healthcare and all those things together would make a lot of sense to me.
Well, I think having lived in the US for many years and having been in Canada now for a decade, one big difference that I see is in the US, you have so many large pharmaceutical companies, large vaccine companies with huge manufacturing capabilities. And we generally don't have that in Canada. So I think COVID's a terrible disease. It's hard to find a silver lining, but if there is one, it's probably that it's telling us we need more research on infectious disease, both viruses and bacteria, because bacteria are still a big threat to us with being antibiotic resistant. We need more research. We also need the capability to respond quickly to the next COVID epidemic. There will be another one. It might be a coronavirus like COVID, or it might be influenza. There will be another one. And I think Canada needs the ability to be able to respond quickly at the research level, but also at the manufacturing level. You've got to be able to make the vaccine in order to get it into clinical testing and then to be able to deliver it to the Canadian population. So I think there is a general realization that Canada does need to have more manufacturing facilities, but we need to make sure we do that, because for sure there will be another epidemic like COVID in the future.
I really echo both of those comments that we need a coordinated strategic effort for pandemic preparedness that's sustained. I mean, that's part of the problem, and I completely agree that we might have the ability to design a great vaccine, but we don't have the ability to evaluate it preclinically, or we don't have a full support of that, and we don't have the ability to manufacture large numbers of doses. And I think considering in a pandemic situation where borders might be closed and supply chains cut, we'll need to be able to really think about, can we do this here in Canada? And the CanSino vaccine was a great example of how the vaccine was supposed to be tested here in Canada in phase 1 clinical trials, but it wasn't, and it wasn't able to make it to our country. So can we do that here? Can we design the vaccine? Can we evaluate it? So if we think about evaluation, we'll need containment level 3 facilities with trained staff. So that's why I'm here in Saskatoon, because in Atlantic Canada, there was no containment level 3 to start working on this virus. As soon as the virus came out, I knew that I wanted to start getting a preclinical model going, and knew that I most likely would not have the capabilities to do that in Atlantic Canada because we don't have a CL3. So I reached out here to Vito Intervac and created a collaboration with Daryl Falzerano, who was able to basically culture the first clinical isolate of SARS-CoV-2 here in Canada. And together we were able to establish preclinical models of SARS-CoV-2, which is now being used to evaluate our homegrown Canadian COVID-19 vaccines. But really, when I look around the country, I don't see many of us who have that capability to do preclinical modeling and in a CL3. So we need both the facilities to do that, plus trained individuals for that, and to have that sustained. And then, of course, we want to, again, scale this up for human use here in our country. And I don't see good coordination between scientists as well with companies the way it is done in the US. So I've basically been sustained with my research program evaluating novel vaccines because of the coordinated efforts that the NIH has, where they match research scientists with companies who are able to provide a novel vaccine, which I don't see here in Canada. And if we had that, I think that we might be able to move vaccines from preclinic to greater use faster.
Well, I do want to say that the Vaccine Task Force is looking at this as much as we can and made strong recommendations around this, because we do realize this is a weakness in Canada. When you looked at around where we could produce the vaccines in a large amount, there's not many places that we could do this quickly compared to other countries. And you're getting vaccine production in smaller countries and the world in this. It comes back to Canada looking at how we go from research, which we've done lots of research and we do some good research. And that's what, when they evaluate Canada's research, they say this, but we don't do well in commercialization. Well, we've got to put things in place so that there is a path to commercialize things easier in Canada than it is at the present time. I go back to the very simple issue of patents. None of our grants will allow us to put any money into patents. And both provincial and federal, we've been told you can't use any of that money for patenting. So how do you really rationalize that you want to see more innovation in Canada when you want to take that money out of patenting? If you don't have the patents, you're not going to get the partners and you're not going to get the development of innovation in Canada.
To conclude our session, I'd like to ask each of the panel members to comment on whether there's anything more that Canada should be doing as a public sector to ensure that Canadians have access to vaccination and therapeutic solutions for COVID-19.
I think that Canada has made a pretty good response. And I think that they're putting the money into, and we should be getting vaccines in Canada. I hope that they'll be timely. We've got some agreements already in place that we should be close to other countries getting the vaccine into Canada. And so I think that's all fine. I'm more concerned about the long-term sustainability of this type of an effort or sustainability of pandemic preparedness in the future.
Yeah, I had exactly the same comments. Very impressed with how Canada has responded and put resources towards this problem, which is great. But we do have to keep this up. This is not going to go away. And I think the example of SARS first time around is clear to everybody now. We should have sustained our preparedness, and then this wouldn't be such a big problem for us. So I totally think Lauren's right on with that comment.
I definitely agree with sustainability of our efforts, but also right now, I think good communication between what's going on in the regulatory realm of vaccines and the general public, so they have an understanding, so they want to take the vaccine, and then also support of those people who are giving the vaccines, so the nurses and the pharmacists, making sure that they have a place to go to have their questions answered.
Pretty much the same. I think Canada has responded very well to COVID. The government has procured vaccines from leading groups around the world. Just an addendum on that, it may well be that repeated boosting is going to be required. We don't know yet, but it may well happen that we need to boost people on an annual basis, perhaps, to keep their immunity up. So that means the government will need to purchase more doses than what they've done already. But I think the way Canada's responded so far has been excellent. And I agree with my panel members. COVID is a wake-up call to all of us, and we need to prepare for the next epidemic. We need to put some things in place so that we can respond better to the next pandemic. But I think so far Canada's done a good job.
Okay, on that note, I'd like to thank all of the panelists for your time and in addressing these important issues. Thank you to the audience as well. And thank you to the organizers, IPAC. This now concludes our session.